This evening, against the background of qualitative descriptions of drug-eluting stents and the unmet needs in interventional cardiology, I'd like to share with you some of the quantitative data from the NERIUS and BIONICS programs, and specifically, too, to share with you some data that we haven't yet presented publicly with regard to the angiographic outcomes from the BIONICS clinical trial. The reticulocyte-eluting EluNIR stent really comprises, I would say, 5 distinct novel attributes, specifically the drug reticulocyte that we'll discuss briefly in more detail, but also a thin strut cobalt stent design that has variable strut width, as previously introduced, that permits more uniform drug distribution under more variable vessel conditions. Aside from the enhanced delivery, stent delivery system to improve performance and deliverability of the technology itself, the elastomeric polymer, too, is unique compared with other biopermanent polymers that are commercially available, given that it confers greater resistance to webbing, bonding, cracking, peeling, and other distortions that so too have been commonly described with commercially available biopermanent polymer drug-eluting stents. And further, and finally, there's a unique manufacturing of this technology. Put simply, a thin sheet of cobalt alloy is laser cut, then sprayed and coated with drug and polymer, and then cut again and rolled into a cylindrical shape, and at discrete points laser welded to create the stent itself. And compared with conventional historical methods, this is a very unique, novel method of manufacturing, and particularly it not only confers greater efficiency in manufacturing, but greater reproducibility, less variability that is so commonplace to drug-eluting stent manufacturing. And this too may introduce or confer substantial cost savings in the device technology. As I've also implied, the drug reticulocyte is a novel sirolimus analog. It has, compared with most other limus analogs, a higher therapeutic to toxicity margin, and very similar, as demonstrated in the left-hand panel, very similar to commercially available drug-eluting stents, including the Xience everolimus stent and the Resolute zotarolimous eluting stent. The drug elution of this stent program occurs approximately over a 180-day period. But in comparison to commercially available stents like Resolute in the right-hand panel, there is no rapid burst or peak concentration of drug immediately after stent implantation, followed by a trough or slow diffusion of drug. But instead, there's a persistent yet low level of drug that is maintained throughout the entire period, the temporal course of drug elution that is identified within the vessel wall over approximately a 3-month period. Both the NERIUS and the BIONICS trials represented a very novel and ambitious both regulatory and clinical trial design program. Unlike historical studies that Alex Sabazed and Eberhard and Marty and Greg and many others have led, in which a first-in-human trial is performed, followed by a secondary proof of concept confirmatory study, led then after by a large pivotal clinical trial, the NERIUS and the BIONICS trials represented the first-in-human experience with this particular drug-eluting stent, but also the first pivotal trials for regulatory approval in their respective geographies. And so the first of these trials, the 2 trials began almost simultaneously, was the NERIUS study. NERIUS was performed in Europe and Israel. Many of you are investigators for this successful program. And in this trial, 302 patients were randomized in a 2-to-1 fashion to the EluNIR stent versus the Resolute drug-eluting stent. The study's primary endpoint was angiographic in-stent late lumen loss at 6-month follow-up, with important attention to clinical outcomes. And the study did achieve its non-inferiority endpoint. The late lumen loss values were exceptionally low at 6-month follow-up, 0.04 millimeters for the EluNIR stent and 0.03 millimeters for the Resolute drug-eluting stent. And these surprisingly low and yet very favorably low late loss values were also confirmed by the Cardiovascular Research Foundation's Angiographic Core Lab by performing 2 separate different types of analyses to confirm these low late loss values. And further, at 6 months, the clinical outcomes also paralleled the very favorable angiographic endpoints, and specifically the rate of target lesion revascularization was only 1 to 1.5%. Now further, with regard to other secondary endpoints, binary restenosis in this trial at 6 months was also favorably low and statistically similar to the Resolute stent, only about 3 to 3.5% at 6-month follow-up. And more recently, Peter Smits, the principal investigator from the Netherlands of this component of the EluNIR program, Nereus, presented the 12-month clinical outcomes for this trial. Again, notwithstanding the sample size of 302 patients, the study demonstrated parity with regard to outcomes of target lesion failure, cardiovascular death, repeat target lesion revascularization, and myocardial infarction related to the target vessel. And more specifically, although not statistically significant, there was almost nearly half the event rate of target lesion failure at 6 months compared with the Resolute drug-eluting stent. But importantly, there was only one occurrence of stent thrombosis among patients treated with the EluNIR stent, and the rate of target lesion revascularization was less than 3%. These very favorable findings in parallel led to the conduct of the BIONICS trial. Now, the BIONICS trial was a very large pivotal trial, 1,919 patients, and it was a study conducted at 8 countries and 76 centers worldwide in Europe, Canada, the United States, representing North America altogether, as well as Europe. And it was a study that randomized patients evenly, 1-to-1, to treatment with either the EluNIR stent or the Resolute drug-eluting stent. And the primary endpoint of this trial was target lesion failure at 1 year of follow-up, but also a subgroup of patients undergoing both angiographic and imaging with IVUS follow-up at 13 months after index revascularization. Importantly, in this trial too, the study as a non-inferiority trial estimated an event rate of 5.3% with a delta of 3.3%. 5.8% rather. And specifically with the BIONICS trial, this study represented what we might describe as a more Commerce clinical trial design. That is, the trial specifically included a much broader, more complex patient population with regard to clinical indications, with regard to lesion anatomy, compared with historical pivotal trials of drug-eluting stents. Specifically, the trial enabled or permitted enrollment of patients with non-ST elevation myocardial infarction, recent ST elevation myocardial infarction, but also patients with lesion complexities that ranged from chronic total occlusions to saphenous vein graft disease, to bare metal stent in-stent restenosis, to bifurcation disease, to heavily calcified lesions that would require atherectomy. Historically, again, all lesion complexities that are routinely excluded from pivotal drug-eluting stent trials. Indeed, the only key exclusion criteria for the study were patients with shock, patients with advanced left ventricular dysfunction, or patients who might not be capable of adhering to a prescribed 12-month regimen of dual antiplatelet therapy. As I've shared, the study enrolled 1,919 patients at 76 centers across 8 countries. And in particular, it's notable that a large number of patients were enrolled in Israel, the United States, Canada, but also the Netherlands and Spain. And enrollment occurred over a relatively rapid time period for such a large clinical trial, approximately 16 months of timeline for enrollment. It's important also to recognize the contributions of many of you all in this audience tonight, investigators in your research programs who oftentimes enrolled more than 100 patients contributing to this trial, so quite remarkable enrollment, specifically in countries of Israel, in Spain, Belgium, the Netherlands, Canada, and in part the United States as well. Over 4,200 patients provided informed consent for enrollment into the BIONICS trial, and ultimately, as previously described, over 1,900 patients were evenly randomized to the EluNIR stent or the Resolute drug-eluting stent. And through 1 year of follow-up, completeness of follow-up for clinical endpoints was achieved in more than 98% of the study population. And further, a smaller subgroup of patients, as previously introduced, underwent angiographic follow-up at 13 months, as well as intravascular ultrasound imaging. And at least previously, as we shared these data at the TCT meeting as a late breaker, and presently a manuscript is under preparation, that we had not yet presented the angiographic and IVUS outcomes that I'll share with you this evening. Overall, however, the study patient population represented the broad inclusion criteria of the trial protocol. Specifically, nearly 1/3 of the study population was diabetic, and over 40% of the patients presented with acute coronary syndromes. But even more remarkable is that approximately 35% of the study population had presented with elevated biomarkers consistent with non-ST elevation myocardial infarction. And further, perhaps more unlike previous clinical trials of drug-eluting stents, there was a high rate of prior myocardial infarction of roughly 30%. While the left anterior descending artery was the most commonly lesion treated in this study, moderate and severe calcification was quite prevalent, approximately 25%. And there was, in fact, a statistically significant difference between the two treatment cohorts in severe calcification, which was more common among patients treated with the EluNIR drug-eluting stent. Bifurcation lesions also were commonplace, representing nearly 28% of the study population. Osteal lesions, chronic total occlusions, vein graft disease, and other were also included. The average lesion length in this trial was approximately 18 millimeters. The average stent length was approximately 24 millimeters, and overlapping stents occurred in about 25% of the study population as well. Overall, procedure and lesion success and even device success were considerably high in this clinical trial and quite uniform, although there was a modest but statistically significant difference in device success between EluNIR and Resolute, but note this did not translate into differences in lesion or procedural success. And with regard to the primary endpoint at 1 year of target lesion failure, which represented the composite of cardiovascular-related death, myocardial infarction associated with the target vessel, and ischemia-driven target lesion revascularization, as a non-inferiority trial, the study met its primary endpoint, but particularly with numerically identical event rates of 5.3% for the EluNIR cohort and 5.3% for the Resolute drug-eluting stent cohort, again achieving the non-inferiority primary endpoint. And those results are simply represented graphically in this figure, simply demonstrating that there was a modest numerical excess, but not significant excess of events occurring with the Resolute stent, largely through 6 to 9 months of follow-up, meeting parity of outcome at 12 months follow-up for the two stent cohorts. Further, with regard to the individual endpoints that comprise target lesion failure, the rates of cardiovascular death were exceptionally low despite the patient complexity enrolled in this trial. Similarly, target vessel-related myocardial infarction was only approximately 3% for both groups, according to a broadened SCAI criteria of myocardial infarction. And what's especially noteworthy is that despite the patient and lesion complexity that I've shared, the rates of target lesion revascularization at 1 year follow-up were only 3% for EluNIR and 2.5% for the Resolute drug-eluting stent. Moreover, we compared myocardial infarction by more varied, broader definitions according to clinical trials criteria, and simply demonstrated no significant differences in the rates of myocardial infarction between the two stent types. What's also especially important to note is that the occurrence of stent thrombosis, despite complex indications with a high prevalence of myocardial infarction, high degrees of patient and lesion complexity as previously introduced, that the rate of definite and probable stent thrombosis through 1 year of follow-up was exceptionally low, only 0.4% for EluNIR, 0.6% for the Resolute drug-eluting stent. These findings were observed against the background of about 75% of the patients adherent to dual antiplatelet therapy at 1 year of follow-up. And what's also particularly noteworthy is that there were no occurrences of stent thrombosis in the EluNIR group after the first 30 days of the index revascularization. And as I've previously shared, we had not, at least today, presented the angiographic and eDES outcomes that for the first public presentation this evening, I'll share with you this evening. So specifically, 85 patients in the EluNIR cohort underwent angiographic follow-up. In addition, 55 of these patients underwent intravascular ultrasound follow-up. And in the Resolute cohort, 73 patients underwent angiographic follow-up, and 56 of these patients underwent eDES follow-up. And at 13 months following index revascularization, Once again, the results achieve non-inferiority and are numerically quite similar. Specifically, in-stent late loss was 0.22, very competitive and favorable for the EluNIR drug-eluting stent, and 0.23 for the Resolute drug-eluting stent. Further, binary restenosis rates were in the single digits for both stent cohorts, and by intravascular ultrasound, again confirming effective suppression of neointimal hyperplasia, the percent neointimal hyperplasia was again in the single digits for both the EluNIR and the Resolute drug-eluting stents, once again also achieving non-inferiority. Further, among subgroups of topical interest relative to geography of enrollment, relative to age, relative to sex, relative to presentation of an acute coronary syndrome or not, we observed no statistically significant differences between the EluNIR stent and the Resolute stent that would favor one stent type versus another. So in summary, from the NEREUS clinical trials program, there was a very low 6-month late lumen loss in stent and in segment and low binary restenosis rates that translated into very favorable clinical outcomes that have been reported and observed through 12 months follow-up. Further, in the BIONICS trial, performed almost simultaneous to NEREUS, the EluNIR stent was statistically non-inferior to Resolute for 1-year clinical outcomes in what we've described as a less selected, broad, more comers patient population. And indeed, the rates of target lesion failure were identical for both stent types. Further, there was a very low stent thrombosis rate observed for both types of drug-eluting stents, and specifically no events for the EluNIR stent beyond the initial 30 days of index revascularization. And moreover, these rates of target lesion revascularization of approximately 3% for EluNIR, despite the patient complexity, are consistent with those observations from the NEREUS trial, both angiographic and 1-year clinical outcomes, but they're also reaffirmed or supported further by the data that I've shared with you tonight of effective suppression of neointimal hyperplasia by intravascular ultrasound and very low late lumen loss values by angiography at 13 months follow-up. So in summary, considering the two programs altogether, both BIONICS and the NEREUS programs represented a very ambitious multinational program, both from a clinical trials structure as well as from a regulatory perspective that combined first-in-human experience with pivotal registration applications. Further, these findings endorse the safety and efficacy of the EluNIR drug-eluting stent in patients who are treated in real-world clinical practice. Thank you again.